A very warm welcome to all my dear students and welcome to yet another super cool, super awesome and absolutely amazing session of Biotechnology Principles and Processes lecture number 1. So this is a very important chapter for NEET where you can expect nearly 5 questions. Okay, so tally up to a total of 20 marks. Sorry, tally up to a, there is some technical issue I think. Tally up to a total of 20 marks. Clear? So let's move with this beautiful chapter. Okay, let's move on. So first is biotechnology introduction. Okay, what is biotechnology? Biotechnology. The biotechnology refers to the use of microorganisms. Use of microorganism, either it may be animals, plant cells, or their products. Okay, either the microorganisms or animals or plants or their products can be used to generate different product at an industrial scale, to use different product at an industrial scales, and that is to be useful to human beings. Very important, that is to be useful to human beings. Suppose if a country wage a war against another country which have a personal grudge using techniques known as bioweapons, that is introduction of a set of microorganisms which can destroy the entire what we can say, destroy the entire country. Okay, bioweapons, use of microbes for attacking another country, use of microbes for attacking another country. So those cannot comes under biotechnology. Biotechnology is something related to the production of services and products in industrial scale useful to human beings. Very important is the term useful to human beings. Then old biotechnology methods are based upon the natural capability of microorganism. For example, formation of citric acid. Formation of citric acid is from a fungus. Citric acid formation helped by a fungus, and the name of that fungus is Aspergillus niger. It is Aspergillus niger. Aspergillus niger. Okay, Aspergillus niger. Next is production of penicillin by Penicillium notatum. Penicillium notatum is also yet another fungus. Penicillium notatum is also another fungus. Clear? Then the penicillin discovery is being credited with 3 people. That is, the person who just actually invented it was Alexander Fleming. Who just invented it was Alexander Fleming. Okay, Alexander Fleming. And his works were, and three persons were awarded the Nobel Prize for the discovery of what, penicillin from Penicillium notatum. And the other persons who just won the Nobel Prize for this was, number one, it was Ernest Chain. One scientist, it is Ernest Chain. Okay, the next scientist is Howard Florey. Howard Florey. Okay, these names of the scientists you will be studying in the chapter Microbes in Human Welfare. Okay, Alexander Fleming, Ernest Chain, and Howard Florey. These 3 scientists contributed to the formation or discovery of penicillin from Penicillium notatum. New biotechnology. Okay, old biotechnology was based on the natural capability of microorganism, whereas new biotechnology techniques are based on recombinant DNA technology. Okay, what it is? A recombinant DNA technology. I'll show you a beautiful jiffy now. All of you just wait for that. 1 second, let me take it off. Yes, this is a beautiful jiffy. Okay, the DNA are being cut down by some kind of an enzyme. Okay, one kind of an enzyme is just cutting down the DNA into fragments. Okay, next is, this is how a particular enzyme just cuts the DNA into fragments. Clear? So let's continue our class. Okay. So new biotechnology is based on recombinant DNA technology, recombinant DNA technology. For example, human gene producing insulin. Insulin is produced artificially, right? Insulin is produced artificially using biotechnology, right? You will study about this in your next chapter, that is biotechnology and its application. You will be studying about the preparation of insulin. Okay. So the preparation of insulin was first carried out by an American company, and that was Eli Lilly. Okay, American company prepared insulin, artificial insulin for the first time. So initially, in the initial production, artificial production of insulin was done before that also, before the introduction of biotechnology also. But the insulin, artificial insulin was prepared from the slaughtered sheep, cows, etc. like that kind of animals. Okay, it caused several type of allergies in certain organisms. As a result, this is transferred to biotechnological method, which prepares taking a fragment of DNA. By taking this is a fragment of DNA that prepares insulin. So this DNA fragment is cut down. Okay, this DNA fragment is cut down, and this is a normal gene of a particular person, and this is the insulin producing gene. This insulin producing gene, this insulin producing gene will be inserted inside this DNA of the host. Clear? Okay. So the vector used over here is E. coli. This is a bacteria. E. coli is a bacteria, and E. coli is used as a vector in all biotechnological application methods. And the another one is a modern biotechnology. Okay, modern biotechnology, we get valuable products. We get valuable products. The main use of biotechnology is in case of medicine. Okay, is in case of medicine. Okay, produced with the help of microbiology, then biochemistry, then tissue culture, chemical engineering, genetic engineering, molecular biology, and immunology. You can see. Clear? So let's move on with the next slide. Okay. Okay. So these are the applications of the biotechnology. First one is a microbe mediated process such as making of curd, bread, wine, etc. are microbe mediated processes. So biotechnology is the first application is it is used in microbe mediated processes such as making of curd, bread, wine, etc. Second one is the correction of defective gene. Okay, the correction of defective gene is yet another application of biotechnology. Third one is the preparation of DNA vaccine. Preparation of DNA vaccine. Fourth one is IVF program, that is in vitro fertilization programs. Also, you will use this biotechnology. And the last one is the gene therapy. Okay, in gene therapy also you will use the technique or the principles of biotechnology. Clear? Next is, okay, genetic engineering. You can expect questions for your board exam also from here, as well as for your NEET exam. So what is genetic engineering? What are the other terms for genetic engineering that we use? Genetic engineering is also known as recombinant DNA technology or gene splicing. Okay, recombinant DNA technology or gene splicing. For what it is done? It is done for the manipulation of DNA. That is alteration of DNA. We do it for the alteration of DNA. Alteration of DNA. Okay. With the isolation, we will just isolate some useful genes from variety of sources, and we will lead to the formation of a recombinant DNA. We would lead to the formation of a recombinant DNA. For example, I'll just show you. Okay, this is how the genetic engineering is just carried out. Clear? So I'll just show you. Okay, let me erase it. Yes. Yes. For example, this is a particular gene in the human body. Okay, and you can see this particular up to here, it just synthesis for what? This part of this particular gene codes for a particular enzyme or hormone, say to be enzyme A. Okay, and another organism B, this B is an organism. Okay, B is an organism deficient in the enzyme A. It is deficient in the enzyme A, so we have to just make this DNA should be incorporated into the organism B. So what should we do? This is the DNA for organism B. We should just, first of all, we remove this particular position. We just remove this particular small fragment of DNA which codes for the enzyme A from the organism A, and we just insert it into the gene of organism B using several techniques are available. If it is a plant, it is with the help of biolistics, or you can call it to be gene gun. You will be discussing about these detail in this chapter. Gene gun means using a tungsten or platinum as what metal. They will be just incorporated or fired into fast moving high velocity tungsten or gold. They may be what inserted into the plant cell. Okay, next one another technique is micro injection. Micro injection. What is micro injection? Micro injection means you have to be just taking out that gene. You will be splicing of that gene that codes for the enzyme, and you will be just injecting it directly into the nucleus of the needed organisms. Clear? Okay, let me have. There is some technical issue. Let me correct it off. Okay, 1 second. Yes. We should move with the third one, right? Yes. So, genetic engineering is also referred to as a recombinant DNA technology or gene splicing used for the manipulation of DNA. Okay, here we will be forming a recombinant DNA. The new DNA that is formed will be known as a recombinant DNA, which can be used for repairing, improvement, and perfection and matching of the genotype. Genetic engineering can be defined as what? Genetic engineering can be defined as a chemistry that alters the chemistry of the genes. That is, either it should be RNA and DNA, and genetic engineering would change what? The phenotype of that particular organism. The phenotype of that particular organism. Genetic engineering may be defined as a technique for artificial of modifying DNA. It can be DNA or RNA also. You can genetically modify it to suit human needs. Clear? So let's move with the next one. Yes. In genetic engineering for manipulation, breakage of DNA molecule occurs at two desired places with the help of enzyme restriction endonuclease. 100% MCQ part. Okay, restriction endonuclease. For example, you can see they comes under the breaking enzymes comes under a category of enzymes known as nucleases. Nucleases are of two kinds. Okay, let me take the board. Okay, nucleases enzymes are of what? Nucleases enzymes are of two types. Mainly nucleases enzymes are of two types. Okay, what are they? First one is endonuclease enzyme. First one is endonuclease, and the second one is exonuclease. Clear? Endonuclease and exonuclease. Suppose this is the DNA fragment. In the higher class, we'll draw DNA just as a line. Okay. If this is the DNA fragment, I'll be writing the code G, A, A, T, T, and C. Okay. If in the case of endonuclease, what will they do? In case of endonuclease, these endonuclease will cut the DNA at specific what? Specific site. Specific sites. That is a function of endonuclease. Suppose in case of exonuclease, G, A, T, T, C is one DNA segment. Okay. And what is the function of exonuclease? Exonuclease just cuts the DNA from ends. Just cuts the DNA from ends. Okay. So they do not have a specific site for DNA sequencing. They do not have a specific site for DNA sequencing. Clear? So I'll just show you a beautiful jiffy upon this. Okay. Okay, we have certain restriction enzymes. Okay, restriction enzyme, which you may be calling it as the molecular scissors. Okay, the restriction enzyme you can call it as to be molecular scissors. Clear? Molecular scissors, that is restriction endonuclease or exonuclease to isolate. So before that, I'll show you a beautiful jiffy. Okay, enjoy. Okay, I'll just show you the function of restriction enzymes first of all. Okay, yes, this is gonna be. Let's watch. First of all, the enzymes are just cut down. Okay, first of all, the action takes place by the restriction endonuclease. The action takes place by the enzyme restriction endonuclease. Restriction endonuclease, because it just cuts the DNA at specific sites, right? I think the color is not visible. It cuts the DNA at specific site. That is including 6 base pairs. That is, the site is called to be what? A recognition site. You will be calling the particular site as a recognition site. So this is first. First, restriction enzyme just acts upon these two positions. Suppose this is to be the restriction enzyme, you suppose. I'll just first show you the action of restriction enzyme. Okay. Which color? Yeah. First of all, this is a restriction enzyme. Okay. A restriction enzyme just acts upon at this two recognition site. And what did they do? They will just break the DNA into two fragments. Breaks the DNA. So 5 dash 3 dash, opposite one 3 dash 5 dash. Sorry. Yeah, 3 dash 5 dash. Here if it is 5 dash 3 dash, next sequence will be 3 dash 5 dash. Okay, so let's see. Is it working? Take it one second. Yeah, it's just joined by hydrogen bonding. Okay, it is just joined. And you have another enzyme, that is DNA ligase. You have another enzyme known as DNA ligase. DNA ligase. What is the function of DNA ligase? It just makes the hydrogen bonding and just welds the bond between what the new recombinants. Okay, it forms covalent hydrogen bond, covalent bonds between the DNA sequences. Clear? So next one. So this is how the sticky ends are formed and it will just join, and this is the enzyme. Okay, this will be the enzyme and it will put bonds, covalent bonds in between. Clear? Yes. So the new DNA molecule is called the new DNA molecule. Okay, the form new DNA molecule will be called the recombinant DNA. And the technique is known as genetic engineering, and the technique you will be calling it as the genetic engineering. Genetic engineering means what? Adding, removing or repairing a part of the genetic material. So there will be two genetic material. First one is RNA, second one is DNA. Either genetic material you can just make. Okay, either RNA or DNA you can make. Clear? Let's move with the next one. Okay, next one. Paul Berg is known as the father of genetic engineering. Very important. Okay, NEET question. 100% direct MCQ for your NEET. Okay. Paul Berg is known as the father of genetic engineering. He just transferred the gene of SV40 virus, simian virus, SV40 virus into E. coli. E. coli will always act as a vector with the help of a phage, that is bacteriophage. He was awarded the Nobel Prize for that. Okay. The concept of genetic engineering was the outcome of two very significant discoveries. Two significant discoveries, simultaneous discoveries, led to the concept of genetic engineering. They were the presence of extra chromosomal materials. Presence of extra chromosomal materials in bacterial cell, in prokaryotic cell. Presence of extra chromatic material in prokaryotic cell, and that you will be calling it as the plasmid, which replicate along the chromosomal DNA of the bacterium. Suppose when it is standing alone, suppose we say this is to be the plasmid of a particular prokaryotic organism. If this is incorporated into a living organism, and this is that incorporated what foreign DNA or foreign plasmid, and you can see inside that organism its natural plasmid was also there, that is the vector DNA. And what happens as this vector DNA replicates, as the vector DNA replicates, this plasmid also just gets replicated with the same copy number, with the same copy number as that of the vector DNA. Okay, then presence of restriction endonuclease, which cuts DNA at specific site. This site you will be calling it as a what? Recognition site. Recognition site, or more advanced you will be telling its name as the palindromic site. Okay, what you will be calling it as to be the palindromic site. That is, what do you mean by a palindrome? Palindrome means if you just call, for example, madam. Madam is an example of a palindrome. If we keep the sequence of reading the same manner, we'll get the oppositely from first to last and from last to first, it reads the same. Exactly like that, the DNA sequence, for example, you can write GAATTC, and that is to be 5 dash 3 dash direction. Then the exact DNA, the similar palindromic site is CTTAAG. That is to be 3 dash 5 dash. So this is an example of a palindromic DNA sequence. Palindrome. Okay. So the restriction enzyme is known as molecular scissors because it cuts the DNA at specific fragments. So this is shown here. This acts as a molecular scissors. This act to be the molecular scissors. Clear? So next one. So this we have already shown the action of what? DNA genetic engineering. Okay, the action of endonuclease and DNA ligase. Okay, DNA ligase. And one more thing, I'll just show you. Okay, so these after the restriction enzyme gets acted upon in this particular DNA, suppose this is to be the 5 dash 3 dash and this is to be the 3 dash 5 dash. Oh, what happens is that it leaves sticky ends. Okay, it leaves behind sticky or free ends. You will be calling it as to be sticky ends or free ends, where you can see the DNA ligase. Okay, this enzyme is DNA ligase. Okay, this DNA ligase just acts upon there to form the hydrogen bonding. Clear? So next one. More details about the restriction enzymes. Clear? Restriction enzymes are also known as molecular scissors. Repeating for several times. Okay, molecular scissors. A large number of specific kinds of enzymes are employed in genetic engineering. First is the lysing enzyme. These enzymes are used for opening the cells to get DNA for genetic experiment. For example, you can see the bacterial cell wall is commonly dissolved with the help of an enzyme known as lysozyme. Okay, the bacterial cell wall is dissolved using an enzyme lysozyme. And in case of plants, we use it as the enzyme is cellulase. We use the enzyme that is cellulase. And in case of fungus, in case of fungi, you will use it to be chitinase. And in case of bacteria, you will use it to be lysozyme. Second set of is the cleaving enzymes. What are cleaving enzymes? These enzymes are 3 types. So cleaving enzymes are of 3 types. First is the exonuclease, second one is endonuclease, and the last one is restriction endonuclease. So all these cleaving enzymes comes under a group that is nucleases. Okay, nucleases. Okay, so let's with the next one. This is what I have already told you, right? So the lysozyme is used for what? The lysozyme is used for bacterial cell wall dissolution. Cellulase is used for plant cell wall dissolution. And chitinase enzyme is used for fungal cell wall dissolution. Clear? Next one. Okay. Exonucleases function we are discussing. Exonucleases. Exonucleases, what will they do? Exonucleases, it cuts off the nucleotide sequences from the ends of DNA molecule. Suppose this is a DNA fragment. Exonucleases will cut. Exonucleases will cut the enzyme from the ends. Okay. Next one is endonucleases. What are endonucleases? They have specific DNA points, okay, including six base pairs. Okay. Next is restriction endonuclease. What are restriction endonuclease? They cleave the DNA at specific position, and what you will be calling that specific positions? We'll be calling that specific position as palindrome site. Palindrome sites, or what you will be calling it as the sequence, origin sequence. Okay, recognition sequence. Recognition sequence. Okay, short single stranded free RNA at the ends. Okay, restriction enzymes E. coli 1. E. coli 1, no, it's E. coli. Okay, it was discovered by Arber, Smith and Nadens. Arber, Nadens and Smith discovered E. coli, and the first restriction endonuclease that was discovered was Hind 2. Hind 2. Clear? Hind 2. From the virus, not from the virus, from the bacteria, that is Haemophilus influenzae. Haemophilus influenzae, which causes pneumonia in humans. Okay, pneumonia bacteria, that is Haemophilus influenzae. And there is a technique for naming all these things. Okay, the naming technique is the first letter stands from the genera of the strain of bacteria from which they are isolated. The second two letters are small. It stands from the species name. Stands from the species name. D stands for the strain of bacteria, the type of strain of bacteria. And the last Roman numeral stands for the order of its discovery. I'll show you a very neat diagram for that. Okay. Yes. For example, I'll show you E. coli 1. Okay, I don't want it to be like this. Okay. E. coli 1. E. coli, that is enough. I have separated each term, the single term. Okay, E. coli, which is a, what is E. coli? E. coli is a restriction endonuclease, and I'm going to explain each term for you. The first letter E comes from the genera or the genus name. The second letter comes from the species name and are represented small. R represents a strain of bacteria from which they are isolated, and I stands for the order. It is not I, it's one. Order of invention or order of discovery. It is a Roman numeral. So it is E. coli. So E stands for the genus, coli, CO stands for the species, R for the strains, and I for the what? Roman numeral stands for the order of its discovery. Clear? So next, beautiful Jiffy. This was a Jiffy actually. Okay, I'll just say. So suppose GAA-TTC. Okay. So the restriction enzymes or the molecular scissors, what will they do? They will just cut the DNA at specific locations, and that is to be the palindromic site, right? Palindromic site, right? It's only here the 3-5' exonuclease function of DNA polymerase 1 and DNA polymerase 2 is being shown over here. So I think it's not working. Okay, next one, beautiful Jiffy again. So this enzyme is what? Restriction endonuclease. This enzyme's name also I was repeating for the 100th time. Okay, restriction endonuclease. And what is it doing? And this is a particular DNA sequence. It's cutting the DNA at specific location. So this cuts the DNA, and as a result, the next enzyme that acts upon here is the DNA ligase. Okay, DNA ligase acts upon here. Okay, just like eating, it just cuts off the DNA into small fragments. Next one. This is also same way. This enzyme is what? Once again, I'll show you. This enzyme is a restriction endonuclease. Every slide I'm just repeating the same enzyme's name, right? In order to make you understand restriction endonuclease. Okay, just be like this. Okay. This cuts the DNA at specific site called a sequence, recognition sequence. Clear? So, restriction enzymes are used in recombinant DNA technology because they can be used in vitro to recognize and cleave within specific DNA sequences consisting of 4 to 8 nucleotides. Usually we call it to be what? Palindromic sequence. This means that DNA sequence is same when read in 5-3' direction on both DNA strand. For example, the term Malayalam. It is an example of a palindrome. Next term, madam. Also an example for palindrome. That is, when the arrangement or the technique of reading is kept the same, it will read the same in both the direction. As a result, the DNA fragments are produced with the enzymes having short, first point, single-stranded overhang at each end. You will be calling it to be the sticky ends or cohesive end. Sticky ends or cohesive end. After the restriction endonuclease have been acted upon, you can see that they leaves behind a particular sequence over there, and that you will be calling it to be what? The sticky ends or cohesive end, where DNA ligase gets acted upon. DNA ligase gets acted upon and joins back the DNA. If I could draw, if it's possible, I'll just make a drawing for you. Okay, very important diagram from your NCRT itself. You guys have to wait because I have a drawing. Okay, this is one. There's another one. So it's two DNA sequence. Okay, and what I'm going to write inside this is the palindrome GAATTC CTTAAG. Clear? Exactly over here also GAATTC and CTTAAG. And this you can be calling, you can call this as to be the what? Vector DNA. This is to be the what? Vector DNA. And this you can call it to be what? The foreign DNA. Foreign DNA. So this is acted upon by the restriction endonuclease, that is E. coli 1. E. coli. Acted upon by E. coli, not E. coli 1. And what is the change that you can observe over here? It is this double stranded. Okay, please don't forget to draw double stranded DNA helix. What you can see over here, it is okay. And over there also you can see, and what happening on the other side is okay. What you can see over here, it is this is G C T T A A. Okay, this is also will be just cutting. Okay, here you can see A T T C and a G over here. I don't know whether I could, yeah, I could erase it. And what we'll be doing over here, it is this will be joined over here by the enzyme DNA ligase. DNA ligase will be joining it over here, and finally the final product that you are going to get over here, it is nothing but it will just look like this. Okay, and what over here it is G A A T T C. One more is there. Okay, one more column. C T T A A G. One more column was there. This should be this one. Okay, it's not white, it's this. So this you will be calling it as to be what? A recombinant DNA. This you will be calling it as a recombinant DNA. Clear? Recombinant DNA, where you can see the parts from both the things. Okay. This is a diagram in a NCERT textbook. NCERT textbook, page number 196. Refer. Okay. So next one. Yeah, it's over. It was over here, and I just simply drawn, right? If you have any doubt, just go through this. Okay, the enzyme just cut the DNA at both the strands. Okay. Okay, the enzyme cuts the DNA strands at the same site. Okay, from here onwards it will just remove. From here onwards it will just remove. Okay. And here also what it will be doing is from here onwards it will remove. And here it is acted upon by a restriction enzyme E. coli, and here it will be acted upon by the enzyme ligase. So this is a very important question for NEET. 100% amazing question. Okay. So let's move with the next one. So another jiffy for you. So this is to be, one second. This is to be what? A wrong one. And suppose this is to be a restriction enzyme. Once again, I'll just show that. Okay. Okay, this is to be a wrong one. Look, they are how they are. They are so sad, right? This DNA sequence. Okay, G A A T T, and here you can write T T G yes. Sorry, G T T G A G. Instead of A, J comes G. So they are wrong. It is a wrong DNA sequence, wrong arrangement, right? It is a wrong arrangement. And what they are going to do is nothing but they are acted upon. This is a restriction endonuclease. Okay, endonuclease enzyme. And what they are, look, what they are going to do for this particular wrong match. They are coming and they are changing that particular DNA nucleotide basis. Clear? So as a result, you have so many diseases just vanishing out because of that particular correction. Okay, I'll just show you or name that disorders for you. Okay, for example, hemophilia, retinitis, Huntington's disease, trisomy 18, muscular dystrophy, Stoneman syndrome, cystic fibrosis, color blindness, sickle cell anemia, Tay-Sachs disease, etc., etc. You can have, you can what clear with the help of this particular technique, genetic engineering, one of the application of genetic engineering. Clear? So next one. This is how the DNA is broken up and is joined by new fragment. Okay. So therefore, what happening over here, it is the cutting of two DNA samples with the help of same restriction enzymes, mixing the fragments with a recombinant DNA molecule can be generated. Enzymes, some especially, cleave both strands of DNA exactly at the same position of the nucleotide in a blunt end or a flush end. In a blunt end or a flush end. Clear? So let's move with the next one. So I think from the next onwards, we'll just start. In our next class, you have these all slides just waiting for you. Okay. So thanking you all. Please don't forget to like, share, and subscribe our channel. Thank you all.